Good morning, class. Welcome back from the previous lessons you have been having. This is teacher Muhindo Augustine, and I'm going to take you through one of the kingdoms of living organisms. If we had to make the review of the previous lessons, we realized that we had talked about the classification tree of living organisms, and we looked at the five kingdom plan, which had kingdom Monera, which consists of mainly the bacteria. We have kingdom Protectista, those are the protists, for example, the animal-like protozoans and the plant-like protozoans. Then we have kingdom fungi, which consists of the fungi and the mushrooms we know. Then you have kingdom plantae, which consists of all these plants we normally see. The green plants we have, and some other types of water plants which are floating on water surfaces. Then we have the last kingdom, which is kingdom Animalia, which consists of animals, all animals. So we had looked at the classification, and I'm going to ask most of the questions as we proceed. So our today's lesson is a subtopic of the classification we had looked at before, and we are going to specifically look at one kingdom, which is the first kingdom, that is kingdom Monera. So when we talk about kingdom Monera, we mean that kingdom which consists of mainly bacteria. And the bacterium cell is in form of an animal cell that we had looked at in the first term. Since this is our second term, we will reflect on what we had looked at. So kingdom Monera consists of mainly bacteria, and these two types of bacteria were classified based on different forms. They can be classified based on their structure. They can be classified based on the number or the colony or the existing, either one single or colony. So we have Bacillus, Micrococcus, and so on. Therefore, when we look at mainly today's objectives, we're going to study the main objective by the end of the lesson. We make sure we understand the basic structure of a bacterium, and we study the importance of each part in the structure of the bacterium. Therefore, when we talk about the structure of a bacteria, we look at it being having the slime layer, which is the outer covering. It is in form of a cell wall, and the slime layer, in between the slime layer we have the cell membrane, and the cell membrane encompasses or encloses the fluid particle of the fluid portion of the bacteria, which we call the cell cytoplasm or the bacterium cytoplasm. So bacteria is differentiated from the rest of the kingdom because for it lacks an enclosed cell nucleus. Therefore, its genetic material is not enclosed in the nuclear envelope, so it lacks a cell nucleus. Therefore, we have to look at the genetic material, which is in form of a plasmid, which is just surrounded within the fluid particle, which is the cytoplasm. Therefore, it has a naked DNA and RNA material. So other particles or other molecules will exist. For example, the glycogen molecules, and the whole slime layer is always enclosed and surrounded by either microvilli, and it has a flagella. And the use of the flagella is for locomotion, and it whips. When it whips, it makes the bacteria to move. Uh, for example, if I look at the use of each part as I had described, we realize that the cell slime layer, the outer covering, protects the inner cells, the inner cell components from damage, and it allows penetration of light. For example, for those bacteria that can carry out photosynthesis, those are the blue-green bacteria, which are always found in the sea. Then the cell cytoplasm, it is the fluid particle that encapsulates or washes other cell organelles or the other bacterium organelles, and the DNA is used during DNA replication during cellular reproduction. For example, bacteria can produce sexually using the pili, which is an extension of the slime layer. And when the pili meet of different bacteria, we see that the positive component is transported or transferred or transmitted to the bacterium cell, which lacks that positive component. It becomes a negative, a positive bacterium, which makes it to be a male. The one which has lost the positive component becomes a female. So there is interchange or exchange of of uh, the sex cells of the bacteria. I think since we have obtained today's objectives, we will maybe make a summary. We realized that today's lesson was about to study specifically kingdom Monera, and we had looked at the structure and we had identified some of the important functions of each part. Then for the rest of the part, I'm going to give you an exercise so you can get down and write this exercise. I'm just going to project on the board and we shall answer it and when we come next time. Thank you.